Hello everyone, welcome to Ritika Tutorials. Today I will talk on the topic Basic Rules of DNA Replication. So now I am going to share my screen with you. So DNA replication shows high accuracy. So we can say it is very accurate and it involves base selection, proofreading and repair mechanism. So how it is very accurate? Because if it selects for the base, if it is incorrect base, it will remove it. So it is having the proofreading activity. Once there is the addition of any incorrect or abnormal base, then it can correct it. And also it correct it by using the repair mechanisms. So we can say that the enzymes which are involved in the DNA replication, they must have the proofreading as well as the repair mechanism. So DNA replication, we can say it occurs with the high degree of fidelity. Now, what are the various enzymes which are involved in DNA replication? In this list, first enzyme is what? Helicases, and they play their role first because they are required for separating the double-stranded DNA molecule into the single strands. So why the separation of DNA double helix is required? In order to make the complementary strands on the separated parental strands. So in order to start the replication process, first two parental strands have to be separated from each other, and this function is carried out by the enzyme called as DNA helicase. So the function of DNA helicase is to unwind or unzip the double strand DNA, and how it does so, it performs its function by breaking the hydrogen bonds between the nitrogenous base pairs in the DNA, and in this process also it uses energy which is derived from the hydrolysis of ATP molecule. So we know that ATP are energy rich molecules, and by its use or hydrolysis, that energy is released and energy is used in the breaking of hydrogen bonds between the nitrogenous base pairs. These helicases are not only required for the DNA replication, but also they are required in the transcription process where the DNA strand is separated and one of the DNA strand is used as a template for the RNA synthesis in the transcription process. So this is a model in order to make you understand how the separation takes place with the help of these enzymes. You can see that there are two strands of the duplex DNA. One is running in 5' to 3' direction and the another one in 3' to 5' direction. Now a bubble-like structure in the central region is created due to the action of the enzyme DNA helicase. How it is created? Due to the breaking of hydrogen bonds between the nitrogenous base pairs. So it will give a bubble-like look when you see it under the electron microscopy. Now this zip-like structure is shown here to designate for the replication fork. So you can see that the replication fork is having the Y-shaped structure here also, and at this side also one more replication fork is there. So two replication fork is there, which means that the replication takes place in both the direction. Hence the replication is bidirectional in nature. And the point where this bubble arises is the point of origin where the replication starts. Now, the next thing is what other enzymes which are required for the DNA replication. In that list, the second enzyme is single-stranded DNA binding proteins. Once the DNA strands separated, then in order to stabilize the DNA, or in other words, I can say that in order to prevent the rewinding of the DNA strands, there are some proteins called as the SSP or single-stranded binding proteins. They will bind to each of the separated DNA strand in order to prevent their rewinding. So you can see that two strands of the DNA, and here the replication process is happening in the separated strands. The SSP protein or single-stranded binding protein in the polymer of 4 proteins it will join to one of the DNA strand and hence will prevent its rewinding with the another DNA strand. So first function is to separate the strand, which is done by the helicase, and next is to stabilize the strand and prevent their rewinding by hydrogen binding, that is carried out by the SSP protein. And the other things in this picture is all you can see here. On one of the strand, leading strand is synthesized, and on the other strand, lagging strand is synthesized. And lagging strand synthesis would be in the pieces, which is called as the Okazaki fragment. For each of the leading and lagging strand, primer is required, and because on the lagging strand, each Okazaki fragment will require the primer, so there are primers for each of the Okazaki fragment. Now here you can see the DNA helicase enzyme is here, it is situated, and it is separating the strands, two strands of the DNA. Now another enzyme you can see here, it is DNA primase on which the RNA primer would be synthesized. Now the next enzyme which we will study is primase. So the role of the primase is to synthesize a short oligonucleotide of RNA primer. So in DNA replication, primase catalyzes the synthesis of the primer, and hence primase is a type of RNA polymerase. So RNA polymerase, what it will do, it will synthesize a short segment of the RNA primer. Now it is complementary to one of the single strand of the DNA template. So one DNA template or parental DNA template, a short region of that template would be complementary to this RNA primer. So the RNA primer will get attached to the small portion of the DNA parental template, and from there the synthesis or polymerization of rest of the nucleotides will start. So at the 3' end of the RNA primer, first nucleotide will attach, and in this way, at the 3' end of further nucleotides, another nucleotides will come and attach. In that way, polymerization of nucleotides, or we can say that complementary strand of the nucleotides will start. Now to the 3' end of this RNA primer, nucleotides are added, and this is added due to the activity of another enzyme which is called as the DNA polymerase, and as its name indicates, its function is to polymerize the DNA, or we can say in polymerization of the complementary strand of DNA. Now next enzyme is DNA gyrase or topoisomerases. Now as the DNA replication proceeds, there is the buildup of some sort of torsion or strain in the DNA. So if that strain or torsion is not removed, then what will happen? DNA replication won't proceed successfully. So for relieving out that stress in the DNA or torsion in the DNA, the enzyme which is helpful is DNA gyrase or topoisomerase. So these gyrases or topoisomerase will relieve the torsional strain that results from the helicase-induced unwinding. So we know that helicase enzyme unwind the DNA. So in that unzipping or unwinding, what will happen? Ahead of the unwinding region, there the stress will be produced, and that stress will be removed with the help of this gyrase enzyme. Now the progress of the replication fork, as we have seen that there are two replication forks which are moving in opposite directions to each other. They will generate the positive supercoils ahead of the replication machinery and negative supercoils behind it. So they first unwind and then rewind DNA strand to prevent the tangling and supercoiling of the DNA. So this can be better understood with the help of this picture. So here two winded ropes are there, and now you can see it is stretched from two ends, and if one of the end is unwind, you can if it is unwinded like this, then what will happen? You can see that the creation of the supercoils will be there, or supercoils will be generated by unwinding the two ends of the ropes. So these supercoils are actually what? They are arising due to the torsion or stress in the DNA, which is created by unzipping or unwinding by the DNA helicase enzyme. So this unwinding will create the formation of the supercoils as a result of the strain. So these supercoils has to be removed in order to carry out the unzipping and replication process successfully. So that supercoiling, removal and unwinding process, the another set of enzyme gyrases or topoisomerases are there. They will act on these supercoils and will relieve the strain in the double helix of the DNA and will aid or will help in the unwinding and replication process successfully. Now, next set of enzyme is DNA ligase. Now, when the replication process is completed, then what the another enzyme ligase is there. They will help in joining the DNA fragments. And how they help in joining the DNA fragments? By catalyzing the formation of the phosphodiester bonds between the nucleotides. So we know that on the lagging strand, there are Okazaki fragments. Now each Okazaki fragment is having RNA primer too. And after the replication, these RNA primers are removed out. Now the gap would be created and then how that gap will be filled up. So those gaps will be filled up by the action of the enzyme DNA ligase, where it will catalyze the formation of the phosphodiester bond between the nucleotides. So as we know that the backbone of the DNA is made by the sugar phosphate and the phosphodiester bonds are there. So those phosphodiester bond will make the backbone. So those backbone or phosphodiester bond will be formed with the help of the ligase enzyme. So in this way, the nucleotides will be joined to each other. In other words, we can say that it will seal the single strand nick between the nascent chain and the Okazaki fragments on the lagging strand. Now another set of enzymes which are there in the replication process is DNA polymerase. So Arthur Kornberg was a scientist who have discovered the DNA polymerase 1 enzyme in the Escherichia coli bacterium around 1955. So there are not only one polymerase, there are many polymerases. And in that list, the first enzyme which was discovered was polymerase 1. Now in E. coli and other prokaryotes, there are 5 different DNA polymerases which were later on isolated. So what are those? DNA polymerase 1, 2, 3, 4 and 5. So there are 5 polymerases which were discovered in the E. coli bacterium. Now these DNA polymerases are having the different functions, and the main important polymerases out of these 5 are DNA polymerase 1, 2 and 3. So what are their functions is summarized or compared in this table. So a structural gene, we know that each of the enzyme, it is synthesized by some gene. So what are the structural genes which are synthesizing these 3 different enzymes? They are also different. For example, Pol A or polymerase A gene will synthesize the DNA polymerase 1. Pol B gene will synthesize polymerase 2, and Pol C or DNA E, it is the another name given in the E. coli bacterium, and it will synthesize the polymerase 3 enzyme. Now these 3 different enzymes which genes which are synthesizing these 3 different enzymes, they are also having different number of subunits in them. Pol A gene will have 1 subunit, Pol B is having 7, and Pol C will have more than or equal to 10 subunits. Their molecular weights are also different. Pol A will have 1,03,000 and Pol B 88,000, and Pol C will have 7,91,500. So molecular weight of Pol C gene is more compared to rest of the two. Now the another activity which is inherent to these DNA polymerases 1, 2 and 3 is their 3' to 5' exonuclease or proofreading activity. So why they are having this proofreading activity? In order to ensure that the replication process is proceeding correctly. If any incorrect base is there, then it can be removed by either 3' to 5' exonuclease activity or 5' to 3' exonuclease activity. So these exonucleases, why they are named so? Because they can remove one nucleotide at a time from the 5' end or from the 3' end. So in this way, the incorrect bases in the DNA can be removed and whole of the DNA will be checked by this proofreading activity of polymerases. Now, out of these three, polymerase 1 is having both the 3' and 5' to 3' exonuclease activity. Polymerase 2 is having only 3' to 5' but not 5' to 3'. And polymerase 3 is having only 3' to 5' exonuclease activity, while 5' to 3' exonuclease activity is absent in polymerase 3 enzyme. Now, polymerization rate, which is represented in nucleotides per second. It means that in 1 second, how many nucleotides will be incorporated into the complementary DNA strand. So that will be designated or in terms of the polymerization rate. So it is 16 to 20 for polymerase 1. It means that 16 to 20 nucleotides are added per second by the polymerase 1. 40 nucleotides per second are added by polymerase 2 and 250 to 1000 nucleotides are added per second by polymerase 3. So you can see that polymerization rate for the polymerase 3 is more compared to rest of the two. Now processivity, which is defined in terms of nucleotides added before polymerase dissociates. What does it mean? That once the replication stops or once the replication is completed, then there is no role of polymerase and that enzyme will dissociate itself from the DNA template. So before its dissociation, how many nucleotides are added to the complementary DNA strand? That is that is investigated or that is calculated by the processivity. So for polymerase 1 enzyme, it is 3 to 200, up to 200 nucleotides will be added in one replication by this pol1 enzyme. By pol2, 1500, and by polymerase 3, it is more than or equal to 5 lakhs nucleotides before the polymerase dissociates. So processivity as well as the polymerization rate, you can see from this table, is higher or highest for the polymerase 3 enzyme. So we can say that the principal enzyme which is taking part in the DNA replication is polymerase 3. Next thing is, now we will discuss in detail about the DNA polymerase enzyme. Now for DNA replication, this DNA polymerase will require some raw materials. So what are those substrates or raw materials for the polymerase enzyme? First thing is that it will require a template or parental DNA strand. Obviously, for a template or parental DNA strand is required for the synthesis of the complementary strand by hydrogen bonding between the nitrogen base pairs, and this base pairing will be governed by the rules given by the Watson and Crick. So it means that A will pair with T and G will pair with C in the formation of the complementary DNA strand on the parental DNA strand. Next thing which is required is a primer. A primer is a short segment which is required in order to initiate or begin the replication or the synthesis of complementary strand. Now this primer will provide the free 3' hydroxyl group, and to this 3' hydroxyl group, the nucleotides will start adding up. So if one nucleotide is added to the 3' of the primer, then second nucleotide will be added to the 3' end of the first nucleotide. So in this way, the polymerization will be proceeded, and that is done, or polymerization is done with this polymerase enzyme. Now, what are these primers? If the question is asked, then you should write about it. These primers are oligonucleotides of RNA, which are having the free 3' OH group, to which the nucleotides are added in the complementary DNA strand on the parental DNA strand. Now to the 3' OH group of the primer alpha P atom. So we know that the nucleotide structure in which the phosphate, sugar, and nitrogenous base is there. So the phosphate is having the phosphorus atom. And if there are 3 phosphate group, then they will be designated by alpha, beta, and gamma. So the alpha phosphorus atom of the deoxynucleoside triphosphate will react and will form a phosphodiester bond. Okay. So the DNTP is what? These DNTP or nucleoside triphosphate, they are actually bringing the nucleotide to be attached to the 3' OH group of the primer. Okay. So the alpha phosphorus atom of this DNTP will take part in the formation of the phosphodiester bond. This we will study with the help of the structure. Let's see what other things are required. Deoxyribonucleoside triphosphate. They will act as the substrate because there should be nucleotides in the cell. If these nucleotides are not there, then how the polymerization or complementary DNA strand would be synthesized? So this polymerase will need following things: template, primer, and deoxyribonucleoside triphosphate. And they will serve one more function of energy provider. This deoxyribonucleoside triphosphate, they will serve dual function. One is of substrate and the another one is of energy provider. The 2-terminal phosphate in the deoxynucleoside triphosphate will have the high energy rich phosphates, and they will give the energy in the polymerization process. Now, this is a fundamental chemical reaction which is going on in the replication process, in which the polymerization of the nucleotides is carrying out or is carried out by this polymerase enzyme. Now you can see it is deoxynucleoside monophosphate whole N plus DNTP. It is giving rise to DNMP whole N plus 1. This is what? Lengthened DNA plus inorganic phosphates. So you can see here, you can understand it better here. This is the template or parental DNA strand. Okay. And this P and this is representing the sugar phosphate backbone. To these sugars, you can see various bases are attached, T, C, A, and G. Now, in order to synthesize the complementary DNA strand, first thing which is required is a primer. So you can see this region, which is bind to with the complementary base pairing to the template, is having the free 3' OH group. So this free 3' hydroxyl group is now ready to attach itself to the incoming deoxynucleoside 5' triphosphate. So these nucleotides should be there as a raw material to get incorporated in the polymerizing complementary DNA strand. So what they will provide? They will provide the new nucleotide to get attached to the 3' OH group. So now this nucleoside 5' triphosphate will bring nitrogenous base as a complementary base pair to attach to the A. Okay, so this is T and it is attaching to the A. Now, what rest of the things, what chemical reaction is going on here? It is having 3 phosphate group because its structure is 5' triphosphate. So 3 phosphate groups are there. So one of the phosphate group will attach itself to the free 3 OH group, free 3' OH group. So this is will form the phosphodiester bond, okay? And rest of the 2 phosphate will be released out in this process, okay? So you can see that this incoming nucleotide has attached itself to the 3' OH group of the primer, and in it has led to the formation of one phosphodiester bond. Now the next nucleotide will be added to this 3' hydroxyl group of this nucleotide. So in this way, the polymerization of complementary DNA strand will take place. Now next thing is, this is a summary of how replication process takes place. So here you can see these are the two template DNA, and the first thing is helicase will separate or unwind the two strands of the DNA. So in the case of E. coli, it is named by DNA B helicase. So DNA B helicase, you can see it is a circular structure of and is comprising of 6 different protein subunits. So it will encircle on one of the DNA strand and will help in unwinding the DNA strand. Now here you can see the green colored, we can say that it is represented by the green color. It is an enzyme called as the DNA topoisomerase 2 or gyrase. In case of E. coli, it will relieve the torsional strain or stress produced due to unwinding by helicase enzyme. Now next thing is, you can see the attachment of the DNA primase enzyme, which will lead to the synthesis of the RNA primer. And the another thing which you can see is the SSB protein, which are required for maintaining the unwinded state and preventing the two DNA strand for rewinding. So it is preventing the rewinding of the separated DNA strand. Okay, so 3 subunits are of each comprising 4 different units of SSB are now here joined to one of the DNA strand. Now this DNA is the DNA on which the lagging strand is getting synthesized. And lagging strand here with the red portion, one Okazaki fragment is shown, and it has formed on the RNA primer. So 3' end of this RNA primer have initiated the synthesis of this Okazaki fragment. Now you can see that its direction of synthesis is from 5' to 3' direction. Now on the second DNA strand, you can see the leading strand is synthesized, and as we know that leading strand is synthesized continuously, so it is, you can see here it is synthesized from 5' to 3' direction, and its synthesis is, we can say that both of the strands are synthesized due to the activity of the main principal enzyme DNA polymerase 3. Now you can see that now the replication fork is moving towards this direction, and the strands of leading and lagging are synthesized. Fork is moving in this direction. Lagging strand is synthesized opposite to the direction of the fork movement, and leading strand is synthesized towards the movement of the replication fork. So in this way, it will proceed and it will complete whole of the replication process. Now the enzymes which I have discussed in this lecture, all these enzymes play their role in the DNA replication in prokaryotes as well as in eukaryotes. So these are common enzymes which are found in both of the organisms. The only difference is that they are named differently, or we can say that you have seen that there are 5 different polymerases in the E. coli, and out of these 5, only 3 enzymes, which are the main enzymes that I have discussed. It is not that the rest of the 4th and 5th enzyme, they are not having any function. They do have the function, but their importance is little. That's why those 2 are not discussed. Now the second thing is that in the eukaryotes, DNA polymerases are there, but they are named differently, not 1, 2, 3, 4, 5. They will be named like alpha, beta, gamma, like that. So in the next few lectures, I will discuss about the replication in prokaryotes and in eukaryotes also.